All right, folks, so welcome. I know we've got some tired people out there. No big deal. We all get tired, myself included. We have 7 school days, if you include today, until spring break. We need to keep pushing. This is not the time to burn out. This is probably the most important unit of the class. So we really need to have a strong understanding of it. We need to be taking notes. We need to be following along in class, following along with the assignments. And if you can honestly say that you're doing that, then sure, you deserve some time to rest. And that time is coming. But I'm going to keep asking everybody to keep pushing themselves, just like I ask myself to push myself, right? So I wouldn't ask anything of you all that I'm not willing to do as well. We're gonna keep going, keep pushing. I really still believe in all of you. I've seen growth take place between the beginning of the semester and where we are right now. We do have more growing to do to get where we want to be. So we just got to keep going, guys. I know we've established some not so good habits, being at home, being able to be quiet, being able to rest, being able to listen to music and watch videos and play video games and eat. Whatever you're doing at home, some of that, is a good thing. It allows you to kind of be more relaxed, but some of it takes away some of your focus in class. We need to get back to it. That's the whole point of us being here in person. All right. Those of you who are at home, hopefully you're sticking with us, you're rocking with us, and we can keep going. So today's lesson, we're going to continue our discussion about inheritance that we started yesterday. I've got a couple practice problems that hopefully will look similar to what you did yesterday as well. Today is March 23rd, 2021, the 3rd day of this 7th unit. We've got the same essential question from yesterday. This time we will be investigating what's called codominance as well as incomplete dominance. All right, so we're tacking on some new structures here today, but we're going to follow the same general mechanism using Punnett squares. Before we get to that, I want to do some quick warm-up review. So I believe this was a question in your warm-up. Should have been a question in your warm-up. It says a strand of DNA reads AAC, CGG, GCA. What would be the amino acid sequence produced from this DNA? Now, some of you may have tried to directly translate this strand of DNA. That's a problem. When you're given DNA, you have to first transcribe it into mRNA, and then you can translate it into amino acids. So the first thing we need to do here is transcribe this DNA into mRNA. What does the letter A get transcribed to? Does anybody remember that? What do we transcribe an A to? Thank you, Rudy. A is transcribed to U. This is a throwback to Unit 5, just before the quarter ended. Okay, so, and then the next A also becomes a U. What does the letter C get transcribed to? G. Excellent. And then, again, another C becomes a G. What about G? What does G get transcribed to? Braylon, do you remember this at all? No? Okay. So here's what I need you to write down if you don't remember this. This is also pretty important. if you don't know, if you're not following along with us on this problem, please write down what you're seeing on my whiteboard right now. A is transcribed to U. T is transcribed to A. C is transcribed to G, and G is transcribed to C. On the left side, A, T, C, G, that's DNA. On the right side with UAGC, that's mRNA. Chandler, you got this? You got this? You didn't bring your notebook. Okay, you still need to take notes. You need paper in that case? Yeah. These are the right width? Huh? Okay. Yes, sir. Real paper. Okay. All right. So Chandler, my next nitrogen base is a G. What does that get transcribed to? No. Look at the whiteboard. Look at your screen. You should be able to see it. You're not in the call? Can you see it? No. Yes, G gets transcribed to a C. And that next G also gets transcribed to a C. The next one gets transcribed to a C as well. What about the C? What does the C get transcribed to? The C is transcribed to a G. Good. And Fredlin, what does the A get transcribed to? U. Good. So when we're transcribing, we're literally going from DNA to mRNA. So and I think you, Chandler and Fredlin and Neftali, if you're writing these notes as well, you should write down this sequence here. those 9 DNA letters. And then under that, you should write the 9 mRNA letters. Okay, now Rudy, can you tell me why do we group them in groups of 3? Yeah, we need to group them in groups of 3 to find the amino acid. Good. And they have a special name when they're grouped in groups of 3. They're called codons. 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 We've got 3 guys here who I want to get some answers from. Okay, can you all see that? A group of 3 mRNA bases is called a codon, C-O-D-O-N. We happen to have 3 codons in this sequence. Rudy, what's the first codon? Not the amino acid. That was correct, but I want to know the first codon, just the group of 3 bases. Which 3 letters did you use to? Good. The very first codon is UUG. What's the second one? BCC. And what's the third one? The last one? BCU. All right. Do you see that? Okay, cool. Braylon, I want you to go first. I know this is going to be, I'm going to walk you through it. What you've got in your hands, and for the folks at home, I've given them codon charts, but you all have the exact same codon chart on your screen. And Natalie and Chandler, I want you guys to go second and third as well. So make sure you pay attention, okay? So Braylon, you're first up. The very first codon is UUG. Now, if you look at that sheet, you can use either side, whichever side you want to use. Let's start with the one you're looking at, though, the circular one. Yeah. What's the very first letter in the codon? U. So we're always going to start off in the middle of the circle. Kind of like a ray of sun. So the first letter is U. So now we're just looking at this section. We're just looking at this section here. Everything else we can ignore. Don't need to even think about anything over here. What's the second letter? U. So now I'm going to move out further. Again, so I'm moving outwards in the circle. So I'm looking at this section here. Everything else I can ignore. What's the third letter? G. So it's a U Good. So now you've translated. At first we transcribed. We had DNA. We transcribed the DNA to mRNA. What you just did was take the mRNA, right? This is you took this mRNA and you translated it to an amino acid. In fact, I'm going to use a different color here. And this is translation. I'm just going to write translation. Okay, Natalia, I want you to go second. So what's the second codon that we have? We use the first three letters. It's okay though. A-L-A. All right, Chandler, you're up. The third codon is C-G-U. What'd you say? G-A. What do you mean? The last codon is C-G-U. So the first letter is C. The second letter is G. And the last letter is U. So what did you end up with? You're confused? Okay. So what's the very first letter of the codon? This is the codon right here. C is the first letter of the codon. Did you write this down? Okay. I asked you to write it down, but it's okay. C is the first letter of the codon. So we're going to start off in this section. You see how I write paper? Start off here. We're moving outwards. So we start off in the middle of the circle and we're moving outwards. So what's the second letter? Huh? Look at this. G is the second letter. So we're moving outwards. So after I started off with C, I can ignore everything up here because I'm only looking in this section. I'm only looking in this section for C. Then you told me G is the second letter. So now I'm moving this way. I can ignore all of this because I'm only looking in the section for G now. And then my last letter is what? U. And so now I need to find this very last letter, U. And which amino acid is that U? What's the first three letters? A-R-G. So that is how you translate. You need to use a codon. These are called codon charts. Again, I want to make sure that you guys are writing this down so you know how to, you can come back to it. You know how to do it in the future. So L-E-U, Fraylin was able to successfully give us that one. Neftali gave us A-L-A correctly. And then Chandler finished it off with A-R-G. So we transcribe DNA first into mRNA. And the second step is to translate the mRNA into an amino acid. That did come up in the warm-up. And guys, I've mentioned this before, but I just want to remind you, excuse me, when you are doing the warm-ups and exit tickets and asynchronous assignments, I almost always will put some type of answer explanation there if you get it wrong. So if you ended up getting the wrong answer on the warm-up, it should have explained how to do this right there for you. Okay, so please make sure you're using that resource. Make sure you're reading the answer explanations whenever you get a wrong answer. All right. Okay, so yesterday we talked about complete dominance. We're going to look a little bit more at it, but today we're also going to talk about a couple other types of inheritance as well. Complete dominance is literally just you get one of two things. You either have the trait or you don't. But in reality, that's not how things work. We can have blended phenotypes as well, which might result in a red flower and a white flower producing to make a pink flower. We could also have what's called simultaneous dominance, basically, where both traits are showing up at the same time. So a black cow and a white cow mate, and they have a spotted cow. That's also possible. So we'll take a look at some of those examples today. Before we do that, I do want to review a little bit with the complete dominance that we talked about. So we did a similar question to this one yesterday. But the question is, for a specific species of plant, green flowers are dominant to red flowers. A homozygous dominant plant is crossed with a heterozygous plant. What are the phenotypic and genotypic ratios of their offspring? Now, there are some big words there. We're seeing some things that maybe confuse us in this question. Please write down the question right now. Just write down the question. We'll take 3 minutes just to write down what you all are seeing on the screen. Franklin, you too. Go ahead and write down the question as you see it on the screen. Folks at home, you all should be following along and writing these down as well. Just write down the question so that you can come back to it. You know exactly what you did. China, can you join the call for me, please? If you do, say it again. You will still be marked present. Yeah. Yeah, so the policy just recently changed, so some teachers might not be doing it yet, but technically you should still be marked present. Okay, so it looks like most of you are done writing. Now, yesterday, stay with me, Naftali, stay with me. Yesterday, we talked about some steps to solving questions like this one. What was the first step? Yes, we need to make a key. That's always going to be the very first step. So your first step is to make a key. And your key should look a lot like this, what you're seeing on the screen right now. We've got 3 columns, 5 rows, dominant, recessive, homodom, which is short for homozygous dominant, hetero, which is short for heterozygous, and then homo recess or res, which is short for homozygous recessive. All right, so I want to see you writing this down, Neftali, Chandler. Make sure you write this key down. We're about to fill it in. This is the first step. First, make a key. All right, looks like we've got the key done. Now we need to fill in the key. So I think in this question, I used the letter F in this question. So Rudy, help us out. I know you've got some good knowledge about this already, so you're going to get the momentum going. The dominant allele, if we're using the letter F, what does the dominant allele look like? It's going to be a capital F. Excellent. And what does the dominant allele represent? In this case, what trait does it represent? It represents green. Good. So keep filling out your keys, guys. Keep filling out your keys. If we're using the letter F, Neftali, I'm going to push you a little bit. What do you think the recessive allele is going to be? Fraylin, do you know? The lowercase f, yeah. So, Neftali, the dominant allele, we always use a capital letter for that. And then the recessive allele, we use a lowercase letter for. Got it? What do you think the color is based on the question? Red, yeah. And that's why it says in the question, green is dominant to red. So green is the dominant allele, and then red is the recessive allele because it's getting dominated. It's kind of like the less important one. Does that make sense? Okay, cool. All right, Rudy, I'm jumping back to you. Actually, Fraylin, you can do this one. What is the homozygous dominant genotype going to be? Wonderful. 2 capital Fs. Excellent. And then which phenotype does that give us, Fraylin? Green. Excellent. Okay, Chandler, I'm coming to you because I see you back there looking at things that are not your notebook. Homozygous recessive. What'd you say? Yeah, that's why I got to get on you so you can start paying attention. But you know how to do this stuff. You can do this. So look at you see how it says recessive? What letter represents the recessive? It's a lowercase f, right? So And which letter represented the dominant allele? A capital F. Now, Raymond told us that homozygous dominant, two capital F, and F is the dominant. So if a lowercase f is a recessive allele, what do you think homozygous recessive looks like? Two lowercase f's. Yes, excellent. And what color, what phenotype do you think that gives us? The phenotype is like the physical trait that you can see. So what phenotype do you think that gives us? Green or red? Yeah, red. Red, good. Good. So guys, when something is homozygous, what we're saying is that there are two of the same alleles. If it's homozygous dominant, that means it's two dominant alleles. That's what Fralin just told us. If it's homozygous recessive, that means that it's two of the recessive alleles. So two lowercase f's in this example. That's what Chandler just told us. All right, now, Natalia, I'm coming back to you. This one's gonna push you a little bit. I'm gonna give you another hint as well. The prefix homo, H-O-M-O, that means same. Okay. And then the prefix hetero, which we're looking at here. What's the opposite of same? Okay, opposite, different is really what it means, different, but your opposite works too. So what do you think? Wonderful. Excellent. So we're going to have one capital F and one lowercase f. This is what we mean when we say heterozygous. We've got one of each. Now, the tricky part here is which phenotype does it give you? What color does it give you? It is going to be green. Good. Why do you think it's green? Yeah, that's right. So, Rudy, can you kind of put it another way? Why is that one green, the heterozygous? Excellent. Yeah. So this is the dominant. So anytime we see that dominant present, it's going to result in making this green. Anytime we see the dominant, we get this. Make sense? The only time we can get this trait is if we have two of them, because it's recessive, it's not as strong. All right, so the first step, guys, we just did this. You've got to make a key. What comes after that, Rudy? Do you remember? Yeah, now we actually make the Punnett Square. Good. And here's what your Punnett Square is going to look like. You really just need to draw this. Four squares. That's the actual Punnett Square, the box with 4 squares in it. Okay, who do I want to go to now? Let's see, Fraylin's drawn. Rudy, can you tell us what are the two parents in this case? The two plants that we're actually crossing. So excellent. Dominant. And what does that mean? What does that mean for its actual genotype? What is this genotype, Rudy? Good. And then, Rudy, I know you're drawing, so sorry for interrupting you, but what's the other parent? I'm sorry, not Rudy. Neftali, what's the other parent? You know that Rudy just told us. It says a homozygous dominant plant is crossed with what? Yes. Yeah. A heterozygous. Heterozygous. And so, what is the genotype of the heterozygous plant? Look at your key. Good. That's why we make the key. So we can always go back to it. One capital F and one lowercase f. Okay. So now we know what the parents are. We've got information about the parents now. Now we want to actually use that information. Now we want to actually use this information to fill out our Punnett Square. So in this case, I'm going to put parent one on the top. And we were able to figure out from Rudy that parent one has the genotype capital F, because it's homozygous dominant. You still with us, gentlemen? You still with us? Have you been writing this down? Okay. That album, whatever you're listening to, can't be that fire. And then we found out from Naphtali that parent two is heterozygous. And he was able to tell us that a heterozygous genotype is one dominant allele, the capital letter, plus one recessive allele, the lowercase letter. So I'm going to write those into the Punnett square on the left side. Okay. Now that we've written in our parents' information, let me just show that on the screen. We need to actually fill in the Punnett square. How do we do that, Rudy? How do we fill in the Punnett square? Yeah, we do what's called cross it. But what we actually do, I'll just explain it in kind of like simpler terms. We're just going to literally take what's on top and we're going to drop it down. And then we're going to take what's on the left and we're going to slide it over. So watch this. I'm going to use arrows to help demonstrate this. That F is going to get dropped down into this box. This F is going to be pushed over into the same box. And we're going to keep following that same logic. I'm going to drop what's up on top down, and I'm going to slide what's on the left over. Really? Yeah, I think you guys do at least something similar in middle school. Now, you do typically write the big letters, the capital letters go first. Okay, the dominant allele is written first. All right. Once you've actually done the cross, as Rudy says, that's the perfect language to use. You've actually filled in your Punnett square. Now we're ready to answer the question. That's the last step. So what is the actual question, Fraylin? What's the actual question? What are we actually being asked to do? The ratios. Good. Yes. Yes. Good. We need to figure out the ratios. And what this means is I want to figure out how many of each phenotype there are. What are the phenotypes in this question? Green and red are the two phenotypes. So I want to figure out how many of each one there are. But it also asks me for the genotypic ratios. How many genotypes are there? How many different genotypes are there? Oh, you're actually right, but I'm talking about just in general. One of them there are. So first we want to know how many green flowers are there. We also want to know how many red there are. So Naftali, use your key. Use your key. That's why we make it. How many of these 4 offspring, right? We've got 1, 2, 3, 4 offspring. How many of them are green? So how many of them? 4. Yeah, good. And how many are red? None, 0. Good. And then let's see. We've got these 3 genotypes. Chandler, keep losing you. How many of each genotypes are there here? How many capital F do I have? Inside the boxes, 2. How many capital F, lowercase f? Which one is it? 2. And how many lowercase f? 0. Good. So these are your ratios now. This is what we're actually being asked to solve. I'll show it on the screen. There are 4 green flowers. That's what Naftali successfully told us, and 0 red. And when we look at the genotypes, there are 2 that are homozygous dominant. There are 2 that are heterozygous, and there are 0 that are homozygous recessive. We didn't get any homozygous recessive offspring here. Are there any questions about this? Okay. Let's have you guys do one on your own. I'm going to set a 4-minute timer. And I'm going to come around after 4 minutes and see how far you've been able to get. We've got a very, very similar question. The only difference is it says true breeding. Does anybody remember what true breeding is? What does it mean to be true breeding? I'll define that on the board for you. I want you working on this question. True breeding equals homozygous. I think that would be a good idea. So I'll even give you 5 minutes. I'll give you an extra minute to write down the question. True breeding means homozygous. It does not necessarily mean homozygous dominant. It just means homozygous. There are 2 ways to be homozygous. You can be homozygous dominant or homozygous recessive. Okay, so I want everybody working on this question on your own. I'm going to start a 5-minute timer right now. No. Yeah. You got 2 minutes. 3. Yeah, at this point, just go ahead and start working on it. You don't need to write it down. Everything you have so far is correct. Excellent. Good job, Rudy. So true breeding means homozygous, right? There are 2 ways to be homozygous. You could be homozygous dominant, which is 2 capital L's, or you could be homozygous recessive, which looks like what? That's heterozygous. 2 lowercase f's would be homozygous recessive. Come on, Natalia, I know you can get this. You've been killing it today. It's the best day you've had all semester. You can do this. Now you can. Folks at home, we've got just under a minute left, and I want to see some answers in the chat. Otherwise, I'm calling everybody's parents. We just did a practice problem. Yeah, you need to start with the key. All right, folks, my timer just went off. Let me see some answers. Where do we answer the question at? You just send the answer to the chat. Send it to the chat. Let's see. What did you guys get? Okay, you got an answer, but that's not the correct answer. But that's better than a lot of things you could have said. So thank you, Alex. I appreciate that. We're going to walk through exactly how to get the correct answer. I got correct answers from Rudy and Fraylin in class. Does anybody else want to jump in with an answer? Jason, Brianna, Ayana, Abraham, Cindy, Monet, Breon, Christian. Don't think just because you're at home, you're off the hook. Still need to work hard. What do you guys have for me? Okay, let's start working through it. So what's the first thing we need to do? Jason, no. Breon, no. Not sure where you guys are getting these. I'm glad you're at least giving me answers that are, close to what you might expect, but they're not correct. Ayana says you're struggling with the crossbox. Okay, so the Punnett Square, that's where we're starting to get a little confused. Okay, thank you for letting me know. We'll go over that. What's the first thing we need to do, guys? And I want this answer from somebody at home. What's the very first step? You guys, Fraylin and Rudy, you can start working on the assignment. It's called U7D. It should be U7D3. Something dominance. Thank you, Ayana. Yes. The first step is always to make a key. Here's what your key looks like. It's the same key that we basically made the last time, right? We just have different information to put in the key. So in this case, I used the letter H. It really doesn't matter. I saw a lot of people in class were using the letter B. That's fine. Whatever letter you choose is totally arbitrary. We just need to make sure we know what the dominant allele and what the recessive allele look like. So I used the letter H. Let's see. Chandler, if I'm using the letter H, what is the dominant allele going to look like? It's going to be a capital H. And in this example, which trait does that give me? The dominant allele. Which trait does it give me in this example? So look at the very first sentence of the question. Not blonde. What does it say? Blank is dominant to what?
Brown.
Good. Brown is dominant to blonde hair. So brown is the dominant allele. And Brianna, because you just spoke up, thank you, I appreciate it. It's good to hear your voice. What is the recessive allele in this case?
Lowercase h.
Excellent. And which, don't put yourself on mute yet. Which which trait does that give me?
Blonde.
That gives me blonde. Excellent. So lowercase h, and that gives me blonde. Neftali, you're doing so well. Come on, stick with me. We have 10 minutes left. So help me fill in this key, Neftali. The homozygous dominant genotype. In this example, what is it going to look like? Homozygous dominant. What kind of H? Excellent. 2 capital H's. And before you keep writing, what is the phenotype associated with that? Brown. Wonderful. Okay, somebody at home. Ayana, you said you were able to do this chart. You said you did the key. So the homozygous recessive genotype, what is that going to look like? Excellent. Thank you, Ayana. And which phenotype does that give me? You're right. It's 2 lowercase h's. What phenotype does that give me? Excellent. Blonde. Wonderful. And now the tough one. Heterozygous. Who else is paying him? Jason or Alex or Breon, one of the 3 of you.
Capital H and lowercase h.
Thank you, Breon. And which phenotype does that give me?
It's mixed.
No, not mixed.
Mixed. Is it true breeding?
It's not true breeding. True breeding is only when it's homozygous. Can Breon get some help from somebody here? It's still going to be brown. Thank you, Neftali.
Oh.
And I'll go ahead and explain that since you all can't hear Neftali. But whenever we have the dominant allele present, in this case, a capital H, whenever we see that capital H, the resulting phenotype is going to be brown. Okay? Whenever we see that capital, even if there is another lowercase h next to it, it's still going to be brown. Okay, that's what makes it dominant. It dominates over that little h. Okay, now we need to make the Punnett square. So the most important information in making a successful Punnett square is knowing what the parents look like. So what is the genotype of the parents here? What's the first parent? And I'm looking for an answer from anybody, Chandler, Neftali, or anybody at home. What's the very first parent? What information does it say about the parents here? What does the question say about the parents in this example? No, it doesn't say anything about them being hetero. The parents in this case, yes, they are both true breeds. Excellent, Ayana. We always know what the parents are because it says that they're being crossed. Chandler, Neftali, stay with me. Come on, guys, 7 minutes. 7 minutes, and then I promise this is the best day you guys have had in biology. Just, you can reap the rewards. But in this case, you see how it says, that's always going to is a true breeding brown bear is crossed with a true breeding blonde bear. That means that they are mating. They're literally having sex. So they are the parents. So they, in this case, are both true breeds. I wrote on the board that true breeding means homozygous. It doesn't necessarily mean dominant, though. So one of the parents is true breeding brown. So what is that parent's genotype? If it's a homozygous and it's brown, then what is its genotype? Good. But remember, we always have two of them, right? So it's two capital H's. Good. Thank you, Ayana. Thank you, Naftali. And then the other parent is a true breeding blonde. So what is its genotype? Chandler, that phone is killing you. I mean, it is just, you can't get away from it. What is the true breeding blonde bear? What is their genotype? Come on, look, use your key, use your key. Two lowercase h's. Thank you, Jason, and thank you, Naftali. True breeding, folks, again, it means homozygous. There are only two ways to be homozygous. You could be homozygous dominant, or you could be homozygous recessive. To be brown and true breeding, you have to be homozygous dominant. To be blonde and true breeding, our key tells us we have to be homozygous recessive. So lowercase h is good. So now we've got the parents' information. Excuse me. Now we need to actually take the information from the top and drop it down. And we need to take the information from the left and we need to slide it over. So we should start to notice a pattern. Natalie, what's going to be in this box? Good. Sammy, what's going to be in this box? Good. And that's how I finish this off here. Excellent. So we find out that they all end up with one capital H and one lowercase h. What is that genotype called when it looks like that? Is it HD, HE, or HR? So use your key, right? This is HD, homozygous dominant. This is HE, heterozygous. This is HR, homozygous recessive. All four of them are one. They're all HE. All four of the offspring are heterozygous. Now we need to actually answer the question. That's always our last step. So I don't want you guys to get messed up and forget what the question is actually asking. In this case, Chandler, what is the question actually asking us? Exactly. We need to know a percentage that are going to be blonde. So, Brianna, I'm coming to you to finish us off. This is going to be perfect. What percentage of the offspring are blonde if we look at this Punnett square? Use your key. Use your key. Which percentage of them are blonde?
Um, 20%.
Okay, so we always have four offspring, right? Do you see the four offspring? Okay, so we always have four. So really that means that your answer can be five things. It could be 0. It could be 25% if there's 1 out of 4. It could be 50% if there are 2 out of 4. It could be 75% if there are 3 out of 4. Or if all of them apply, then you could have 100%. In this case, how many, if we look at the 4 offspring, how many of them are blonde?
2.
Which 2?
Recessive and homo recessive.
Okay, so you're looking at the key. We need to look at the Punnett Square. The Punnett Square is that bolded box that's on the right side of your screen.
Uh-huh.
So when you look at the 4 offspring that are inside of that box, how many of them are blonde?
None.
None of them, exactly. So what percentage is that?
0.
0%. Exactly. Good job. Thank you for being resilient there. So 0% will be blonde. We can't forget to answer the question. We know that because as Brianna just successfully did, we look in the Punnett Square. We see that inside the Punnett Square, all 4 of the offspring are what are called heterozygotes. They've got one capital H and one lowercase h. We can use our key. That's why we spend so much time making the key. The key tells us that the heterozygotes are brown. That means that none of our offspring are blonde in this case. All right, guys, I need to give you some homework. There's no way around it. The homework is going to be, hold on. It's an assignment on Canvas called U7D3. What's it called, Rudy? Are you looking at it? U7D3 Complete Inheritance Practice. Okay, so you guys need as much practice with this as you can get. Please do that assignment. You did it? You finished it? Excellent job. Thank you, Fraylin. Everybody did really well today. I appreciate you. Naftali, I need that same energy tomorrow. You were killing it. You too, Chandler, come back with that same. Oh, you guys aren't coming to school tomorrow. Tomorrow we meet at 9 o'clock, I think, or 8:50. I can't remember, but we meet earlier. So I'll send out a remind message. Have a good day. I'll talk to you guys later. And see you guys on next week, Monday.